welcome everyone. Thank you for joining us this afternoon for this webinar with PPMD and Solid Biosciences. So we're joined today by the Solid Biosciences team to talk through updates to INSPIRE Duchenne, which is their clinical trial with SGT-003, their investigational gene therapy product, delivering a microdystrophin with their novel AAV capsid. As we go through the webinar today, if you have any questions that come up during the presentation or related to the product or the trial, we encourage you to submit those questions to the chat function at the bottom of your screen. We'll have an open Q&A at the end, and we're going to try to get to as many of those questions as we can. We're also recording this webinar, so if you have to step away or if you have friends or family who would benefit from seeing this, it should be up on PPMD's website in the next week or so, and so you can share that as well. But I think with that, we can dive in. I'm going to hand it over to Annie to make introductions and get us started.
That's great. Thank you so much, Eric, and to the entire team at PPMD. We're excited to be back and chatting with the community. I'm Annie Ganote. I'm one of the co-founders of Solid Biosciences, and I lead patient advocacy. I am joined by my colleagues, Pat Gonzalez, who's a senior director in our clinical science team, and also Matt Harmelink, who is a medical director of our therapeutic area head for neuromuscular. So he really is one of our lead doctors in our Duchenne program. Next slide. As a publicly traded company, I have to show our forward-looking statements. I could certainly joke that I ask you all to memorize it and be able to share it later, but I will spare you that. So for our agenda today, we're going to provide an update on our INSPIRE Duchenne clinical program. We're going to take you guys through some of the non-clinical work, an overview of SGT-003, and of course, then we want to dive in deeper into where we are right now with our INSPIRE Duchenne clinical program. Next slide. For those of you who may not be familiar with Solid Biosciences, we founded the company following my son's diagnosis of Duchenne. We founded the company in 2013. We are dedicated to neuromuscular and also more recently to rare cardiac indications. We're based in Boston, Massachusetts, and we partner with advocacy groups and ensure that we have a strong connection to all the patient communities that we serve. Next slide. I'm going to pass it over to Pat Gonzalez.
Thanks, Annie, and thanks to PPMD for the opportunity to really go into the program again and deliver the exciting updates that we have today as we keep moving forward with the clinical study. To kick things off, what I wanted to do was go through a bit of an overview again of what is SGT-003, how does it compare to other gene therapies, especially that are in the works, and really what made us excited about pushing this program forward now for patients with Duchenne. So as mentioned, 003 is a next generation microdystrophin gene therapy for Duchenne. And we'll go through kind of what the different components are that make it so. So to level set again, I think most people are aware of this, but overall at its core, a gene therapy is really there to be able to deliver something that can then replace functionality that could be missing. And so in Duchenne especially, we know that this is caused by mutations in the dystrophin gene and the loss of that functionality. And so as we've moved forward with our gene therapy approach, we use these different core components to be able to now replace what is missing, replace that dysfunctional dystrophin with a different version. And so the key pieces that we'll be talking about here are really the delivery vehicle, and especially related to a next generation approach. This is where is a key focus of our 003 program. The promoter itself, which is going to drive expression, and for our program is going to be constricted to the muscle as well as skeletal muscle and cardiac muscle. And then importantly is the actual transgene that you're delivering and what's going to be replaced, because as we have a large dystrophin protein in its full-length form, we know that we have to make choices into what can be delivered through an approach like this to be able to then have expression of a functional version that can have that functionality systemically. And so kind of taking each of those different pieces, we look at this as the real foundation of what SGT-003 is and why we consider it that next generation. When we look at what that transgene is that's being delivered, the 003 microdystrophin is unique in multiple different ways. a key component of this is the inclusion of the nNOS binding domain. In the next slide, I'll go through a little more detail. But essentially, as we have to look at different pieces that we are including in this overall transgene that we're delivering, we know some are going to be more essential from that piece of having structural stability, but there's a lot more functionality in a dystrophin overall that we know that we need to be able to restore to have the most optimal benefit. And so these are choices that were made to put this construct together. Really a core component of what SGT-003 is this novel muscle tropic capsid called AAV-SLB101. And we'll go through what the details of that are as a reminder, but essentially what we did was go back to understand if we're going to move forward with a gene therapy that's going to have a meaningful benefit, how can we potentially be able to improve on the efficacy and also safety, as we know we're administering very high doses of this systemically to be able to have an impact on patients. And so just from the quick results here that we'll go into a little bit more, we've seen that in changes that have been made to this capsid, we can actually result in multiple fold higher levels getting to the muscle, so to the targets that we want, and reducing levels in the liver. So more to the muscle is a better thing, and less to the liver is a good thing as well, because we want to impact both the efficacy side as well as the safety side. And then coming together and how do we really make this as a therapy so that we can get to patients, be able to make enough of it to have these high systemic doses and to also make sure that we're maintaining purity. One of the things that we really focused on from a company perspective was making sure that our manufacturing was going to be as optimized as possible. And so we've taken this from the side of looking at the full to empty capsid ratio. And what we can actually see here is that when you're looking at more full capsids, when we think about this from dosing patients, we know that more total capsids are going to be a total viral load that could result in a safety signal. If you were to improve that, so you reduce your empty capsids and focus on your full capsids, now you're really delivering your therapeutic payload in a more pure form. And so what we've seen in non-clinical studies is that not only is that going to look better from reducing the total viral load, but we actually have seen that that could also result in improved efficacy as well, just in looking at expression of what we're delivering. So together, this really brings together what our 003 program is, and we'll go through each of these a little bit more in detail. So I mentioned the microdystrophin itself that's a part of this SGT-003 being unique. And that portion here is this nNOS binding domain. And so when we look at a full-length dystrophin protein here at the top, we see that there are many different components. Really, there are structural, as I mentioned, which are going to be able to have that stability for a muscle. And so when you're contracting and relaxing, you have to be able to actually be able to withstand that to prevent the damage to the muscle. When this is absent, that's really what happens is you're unable for that muscle cell or myofiber to be able to withstand it, and you have this injury and overall turnover over time that reduces that muscle function. So when we're looking at a microdystrophin, and all of this has really been based on the work that's been done by Jeff Chamberlain and Dongsheng Duan to understand what are key pieces that are essential for functionality, and we restore those here in a microdystrophin. But now with these many repeats in the center, if we can choose what could bring maybe more functionality than what could come from just a structural sense, we know that can potentially have additive benefit. In the non-clinical studies that were looked at, what was seen is that including these R16, R17 repeats, so what is known as the nNOS binding domain, had actually been able to show prevention of ischemia-induced damage to muscle. So what does that mean? It means that when your muscle is getting exercised, when you're using it more, you need to be able to then supply it with energy again. And that usually comes from the form of blood flow. When you have nNOS that's active at the membrane, you're able to get that better blood flow to the muscle. So then you won't be able to have that ischemia or really lack of oxygen there that can result in injury. And so overall, this is why this construct had been something that we wanted to focus on. And really since the core inception of Solid and microdystrophin programs, we've always focused on wanting to push this specific construct forward. This is also related to different phenotypes that have been seen in Becker muscular dystrophy. So if we look at different mutations, what is or isn't present in a certain patient, we know that there can be a lot of variability in the dystrophin protein that Becker patients do have. But when there has been the inclusion of this nNOS binding domain, there have been milder phenotypes seen. And so overall, this has really pushed us to want to include this for our SGT-003 program. The core of this, as I mentioned, is this AAV SLB101 capsid. So the delivery truck that's actually going to be taking what that transgene is to the muscle. And because this is, as we know, a systemic disease, and we have to be able to actually have a systemic effect. And so with an AAV, we're able to administer this as an IV infusion. So we know that this is something that could be able to actually go through the system and be able to get to all of those target muscles potentially. And so with this selection, what we did was really look at a first generation capsid such as AAV9, and that's really what this was based off of, and wanted to improve the ability of this capsid to target muscle better. And so as we're looking for our target tissues to have better expression, what you can see here in the top panels are that we do see multiple fold higher levels actually getting to the muscle, and this is for both true for both the skeletal muscle as well as for the heart. And then at the same time, while we're targeting muscle better, at that same systemic dose, what we can actually see is that when more gets to muscle, less gets to off-target tissues such as the liver. And so as we know that when you're giving a large systemic dose, you're going to get a lot of this virus that does get to the liver. What we wanted to do is reduce that. And so you can see here with the lower levels in the liver that we've seen about half of the amount getting there. And this is from mouse studies to start, but we've moved forward as well into larger species as we then progress to the clinic. And so some of the first studies that we did were looking at non-human primates. And so looking again at reporters to see, are those patterns that we observe in the mouse studies going to be consistent as we move up in species? And the answer was yes, is that in these studies, we could see in the skeletal muscle, we're continuing to have multiple fold higher amount of actual biodistribution and then expression in our skeletal muscle. The same is true for heart muscle as well. And then again, on the opposite side, we actually are still seeing that we have less getting to the liver. So more efficient on the skeletal and cardiac muscle side, and also more optimal on the off-target side by reducing this overall burden to the liver and off-target tissues. With that, we also wanted to make a comparison to say, if we're really looking at this program and want to understand how does this compare to other first generation approaches that are out there right now, what we want to do is not just look at AV9, but look at other capsids. And so here was a study that we did comparing our SLB101 to RH74. And so RH74 in that clade E is similar to an AAV8 as well. What we can see is when we administer the equivalent systemic dose of an actual microdystrophin construct that's either with the delivery vehicle of RH74 or of SLB101, we can see that we have multiple fold higher levels of actual microdystrophin protein expression with the SLB101 rather than the RH74. And so this is what we saw between both the heart and the quadriceps as well. So about 4 to 6 times higher levels of actual microdystrophin protein expression. And so for us, this is one of those really exciting pieces of data that just showed a reason for us to push this program forward as quickly as possible, understanding that there's always room for improvement, and we want to make sure that what we're pushing forward is something that can be potentially meaningful. So all of that really came to putting us together for our IND-enabling clinical studies that were necessary to then initiate our clinical trial that we'll talk about. And as we can see in these early studies, which we had done both from the mouse and also non-human primate as well, we see that even at these low dose levels here, so 2E12, 6E12 vector genomes per kilogram, we have quite high levels of microdystrophin expression that we're starting to see, and that these are dose dependent as well. And while our microdystrophin has this specific R16, R17 nNOS binding domain, we can also look at nNOS activity and show that when microdystrophin, this specific microdystrophin SGT003 is being expressed, we also have the restoration of that nNOS activity to these muscle fibers. So again, allowing for them to be able to then have the production of nitric oxide when necessary to increase that blood flow to the muscle with energy demand. And so those were the pictures to really show that dose dependency. And as we then look at other tissues as well and quantify this further, what we can see is that even at early days, we have really high levels of expression that are beginning. And so this is looking at microdystrophin percent positive fibers. And then by day 29, really essentially saturated levels out to 100% of positive fibers at all of these dose levels that were tested. So here as low as 3E13, still showing 100% positive fibers across these different skeletal and cardiac tissues. That then is persistent as well out to later time points. And then how does all of that correlate to our function? We always want to test to see is the expression of our transgene actually going to have some association with the functional endpoint? So in the animal studies, what we can look at is overall endurance levels, and this is what we look at here on the left, is really the ability to continuously perform on a treadmill exhaustion test. And we can see is that we have significant improvements, again, even as low as the lowest dose level that was tested. And on the right side, looking at actual direct muscle function. So here looking at a single isolated muscle from the lower leg, what we could see is that force production, whereas it's going to be decreased with the untreated MDX vehicle, so this is our dystrophin negative model of Duchenne, here even at that 3E13, we have really a robust restoration of function back to the wild type level. And so I think this is something to also highlight, is that I think a lot of the times we're trying to understand what's the potential of a microdystrophin. At least from this assessment, what we can see is that we can actually restore back to that wild type functional capacity in this specific force evaluation that was done from these studies. So again, exciting data that we put together here. From there, we always want to look at biomarkers, and this is something that's very important to be able to be monitoring, especially at earlier time points. And so we can see, I think one thing that we all understand very well is serum CK, so creatine kinase levels. And this is something that even at, again, our lowest dose level is significantly decreased back near wild type levels. And that's true of all the dose levels here. Evaluating more recent and novel biomarkers as well is something we want to do to be able to put together a full story of really what are we seeing here and how can we understand disease progression. And so this is serum titin. So titin fragments that are actually in serum and circulation. You can see that holds a similar pattern to what we have from the CK. So two independent markers moving in that similar direction. And then similarly as well, looking at AST here, seeing that overall decrease and a little bit more of a dose response here at those lower levels even. And so we were excited, I think, in our last update to be able to share that our program was cleared for clinical evaluation, and this was first by the FDA, and that was really at the end of last year. Overall, we're excited about the data we've seen so far and the potential for SGT-003 to represent a next generation therapy for Duchenne, really because of that enhanced targeting of our muscle and then detargeting of non-muscle. And so from this, really, we pushed together our initial phase 1/2 clinical study for Duchenne, which is called INSPIRE Duchenne, which is active enrolling. And I'm going to hand it off to Matt now to be able to take us through what is this study and how are we expanding from today onward.
Great. Thank you, Pat. Thanks for having us here. So our INSPIRE Duchenne study is a phase 1/2 clinical trial to evaluate SGT-003. It's to be conducted in the US, Canada, and Europe with a single dose level, the 1E14 viral genomes per kilogram in an open label study in ambulant patients with Duchenne's. And our prophylaxis that we're using is prednisone as immunomodulation. We're looking at two cohorts currently, age 4 to under 7, and then a cohort that is 7 to under 12 with a weight under or equal to 50 kilograms. Both cohorts are currently open and actively enrolling with a target enrollment of 43 total participants. In the trial, our key analysis is focusing on safety at day 360, but we also are looking at microdystrophin expression at day 90 and then the time to rise at day 540. Next slide. As we look at the trial, there are, of course, inclusion and exclusion criteria that are important. So as I mentioned before, we do have these two cohorts, the 4 to under 7 and the 7 to under 12-year-old cohorts. We do have a DMD mutation exclusion for those who have any deletion in exons 1 to 11 or 42 to 45 inclusive. From functionality in the cohort 1, there is not an exclusion or inclusion criteria, but for cohort 2, there are criteria based off time to rise and the 10-meter walk-run test. Patients do need to be negative for AAV antibodies, and they can't have previously received a gene therapy, either investigational or commercial. There is a washout period allowed for exon skipping therapies, vamorolone and or givinostat of at least 12 weeks prior to entering the study. The steroid regime that is allowed is daily oral steroids, either prednisone or deflazacort, for at least 12 weeks prior to entering the study as well. Next slide. Because we are looking at a safety type study, we do have a lot of 30-day initial visits. Really, the question is we want to follow the patients very closely with frequent checkups and blood work to monitor for safety. After that, the visits gradually become less frequent, but they will continue up until 5 years post-administration for the long-term follow-up as required by regulators. Efficacy is looking at 3 total muscle biopsies to be performed during the study with a needle biopsy technique, one at around screening, one at day 90, and one at day 360. This will be used for the evaluation of microdystrophin expression. In addition, testing will occur with different motor function, pulmonary, and other patient-reported outcomes. There are in-clinic testing questionnaires, and then also some at-home video and activity monitoring as well. So a very broad reach of looking at different areas. For the study already, as you've heard, it is active, and so we have achieved some milestones. We had the first clinical site activated in Q2 of 2024. Our first participant was dosed with SG003 in Q2 of 2024 as well. And to date, 3 participants have been dosed by the end of Q3 2024. Right now, we have not had any serious adverse events reported to date after administration of SG003. We did amend the protocol initially to what I mentioned before to have a larger target evaluation for efficacy. If you look at the prior protocol, it was a slightly smaller group and different criteria. So it has been amended and expanded somewhat. Upcoming activities right now are the current active sites are screening and enrolling participants, as mentioned. We are having upcoming active sites, identifying more patients for participation, and we hope to have 90-day data from the first 3 participants shared in Q1 of 2025. Next slide. There's also clinical site expansion underway. So we have multiple additional sites in the US that are in study startup. We're expecting new site activations to begin as early as Q4 of 2024. And we also are expecting 1 additional site to set up in Canada. The clinical trial application has been cleared by Health Canada, and that site is expected to start up again in Q4 of this year. In Europe, additional sites have also been identified for setup in parallel with regulatory activities. The clinical trial applications have been submitted, and we're expecting site activations in the first half of 2025. Next slide. And then here you can see the current status of where new sites are either activated or soon to be activated. So we have multiple sites that are active in the US currently, as well as 1 site in Canada, as mentioned before. And I'm going to give just 2 seconds for people to take a look at the slide, because otherwise we can go through fast just to know where there might be a site near you. Okay. If we can move on then. So I do want to thank everyone. Here again is some of our information that we can stop on, as well as a QR code that will navigate you to the study posting on clinicaltrials.gov. If you have any interest in the study or more information, we do encourage you to reach out to the sites, and there's contact information there. Thank you.
Thanks, Matt. I think at this point, we can go into the open Q&A. So if you have questions, please use the chat function at the bottom of your screen to enter questions there. So we had some that were submitted before, and then I think it might be helpful if we go back a little ways to, I think, like slide 10. Can kind of walk through again some of the information to help make sure that folks are following. So I think what I wanted to ask here, Pat, was so talking about SLB 101 being a novel variant from AAV9. In a previous kind of presentation Solid had done with PPMD, you had a chance to kind of explain a little bit of how you got from AAV9 to this SLB 101. It might be helpful just to kind of talk through what that process is, how you go from something like the AAV9 to this more muscle tropic capsid.
Yeah, absolutely. Appreciate that opportunity to go into detail. So essentially what was looked at was, if we're looking at a first generation AAV, we understand that it has properties, and all of the different serotypes of AAVs are going to be essentially more or less tropic to a different tissue type. When we say tropic, we really mean the ability of a capsid to be able to get to a different tissue type and to be able to then get in and express a transgene that's being delivered. And so we started with AAV9 because we do know that AAV9 has a very good profile for getting to skeletal and cardiac muscle to begin. And there are a couple of different AAV naturally occurring serotypes that are similar. However, what we wanted to see is how can we optimize that even further? So if we looked at this, and this is really going back to the lab and going back to research and understand, if we see the capsid and the properties that we might be able to change that could actually target our muscle tissues better, how much better can we do that? And so what was actually done was taking different peptides, so short actual sequences, adding them to the capsid sequence and seeing if, based on essentially a rational design of choosing these that could potentially bind to different receptors that are present on muscle, are we then able to get more of the capsid to the muscle? And so we were actually evaluating many different of these different peptide insertions. Our non-clinical early stage data were really looking at initially in vitro, so looking at muscle cells in vitro and seeing how different are each one of these different peptide insertions going to be. And what we ultimately decided on was this AAV SLB101, because in looking at initial human Duchenne cell lines, we could see that we had this profile, which was multiple fold higher levels that were getting to the muscle and then resulting in that overall transduction. And that's kind of the first half of the story too. I mentioned that on the liver end, we do get less amount that goes to the liver. The way that we had developed this capsid was not in that we were actively detargeting the liver, we were actively targeting the muscle, but what also resulted as a consequence, as a very beneficial consequence, is that when you do get more on target, you get less to your off target. And that really came together as the full profile that we were hoping for with the next generation capsid.
Great, thanks. I think that's helpful as people kind of try to track these things. It can get a bit complex. I think on the next slide, you had just some of the NHP data. And this is just to, I think, as a reminder for any of the families, looking at this. And I appreciate that you have the animal model symbol in the top corner for each one of these, so it's a little easier to check. But as a reminder, the MDX model is a Duchenne model, whereas these NHPs are not a Duchenne model. If you can kind of just maybe touch on some of the differences there in terms of how you're using those models.
Yeah, absolutely. So when we put together the full package, I think initially what we wanted to do in characterizing the capsid was understand we're ultimately going to be developing this for Duchenne, and this was really how we started off with the capsid. So we use the MDX mouse model because it is dystrophin negative. So we know that from a genetic standpoint, we have an actual model for Duchenne. We also know that we have a functional profile where we can actually look to improve functionality of the mouse model. So a lot of the safety and efficacy can be looked at in that system. However, as we look at safety as we're moving towards a clinical trial, we usually also want to look at a larger animal species. As you mentioned perfectly correctly, is the non-human primates, they are just going to be considered essentially wild type or healthy animals where they are still going to be expressing dystrophin naturally. So we don't always want to look at them for an efficacy profile, but we do look at this to see two really important things. One is, what is the safety profile as we're going into larger animal species, as we're moving towards a human in that direction of being larger? What does the actual immune profile look like? And so this was a key component of putting together our overall package, was wanting to make sure that we have a really good safety margin that we see in these animals. And the second thing is also just, as we're looking at a novel concept here, which is this new capsid, do we have some confidence to show that this response that we saw in the small animal species is going to be scaling up? And so this was really a reason for looking at these specific things. So biodistribution, confirming that that pattern that we saw in the smaller animal species is still consistent. And then also as we looked in as an actual microdystrophin study as well. So this was the separate study that was done to see that that pattern of higher microdystrophin expression was also occurring in these animals, despite having underlying dystrophin. So we're not really looking at this from functional efficacy, but just making sure that the capsid and the payload are doing what they're supposed to be doing.
Then did this kind of inform the dose you arrived at for the trial that won you to the 14?
Yeah, and it was really important for us to, I think, have one very key thing in mind. It's that we know that this is a one-time gene therapy administration. As of right now, we know that redosing is not currently an option. So when we think about what is the dose level going to be when we move into the clinic, we want to not target a minimally efficacious dose, but we want to target something that could potentially be that optimal one-dose therapy. And so I'll actually quickly move over to one of those other studies from the non-clinical as well. But what we're excited to see is that as we put together the total amount of data from a functional side, here you could see we looked at doses here down to 3E13 and actually even lower down into the E12 range. And what we could see is that there was efficacy down there at these lower dose levels. We saw that there was this restoration of function back to wild type levels at 3E13. We also saw that we had microdystrophin expression that was quite high levels as well, even in the E12 range. But I think what we've all learned is that when you come from an animal model to a human, especially for microdystrophin gene therapy, it's not going to be a one-to-one. And so we want to make sure that we're accounting for anything in that scale up to a human that we can. And based on our safety profile actually being very good and at the highest dose level that we evaluate in our non-clinical package, which was up here at 3E14 showing no safety signals, we wanted to really take that approach and want to target something that could provide that optimal level of benefit. And that's why we ultimately chose this 1E14. There could be a lot of data that says that maybe we could have gone a bit lower, but we don't want to undershoot essentially when we're looking for this effect.
That's helpful. Thank you. Actually, I think that also might be a good spot just in case for families who maybe are newer to hearing about gene therapy, because they might hear the term or talk around redosing. Because we always say it's a one-time therapy, and it's not necessarily because we're saying that this is going to be the only therapy that you need. I think often when we think about gene therapy, we think you're still going to need combination therapy. But for this one time, can you just kind of help explain, what is the challenge with redosing or why we're not able to do that at the moment?
Yeah, and I'll even go to just the eligibility for it. And so right now, what we know is that kind of like any other virus that you get when you administer a gene therapy, your body is going to naturally produce antibodies against it. And so because we are looking to take advantage of that virus actually getting this payload to a muscle, to a cell, and then to be able to express this, what we know is that when there are antibodies that are present, they then essentially prevent this treatment from doing what it's supposed to do. So these are the two pieces here for when we look at both the antibody criteria. So for pre-existing antibodies, I think one of the questions is, could there be a potential immune response that's heightened if you do have antibodies? But a bigger one that has been looked at is also from the efficacy side. We do know that there will be reduced efficacy if you do have antibodies. And so that's something that we're trying to prevent. And then with prior treatments here, this is really the reason for not allowing patients who have had a previous gene therapy, because as of right now, we don't have the confidence that it would be able to do what it's supposed to do. I think to your point too, right now for redosing, this is something that many of us are looking into. We really want to see what can we do about pre-existing antibodies? What can we do about antibodies that come up post-dosing? We're not there yet right now. And so this is why these criteria exist, but we really hope to be able to move forward in that direction as well over time.
Yeah, thank you. And I think, it's also, again, there's the safety component of that as well for someone who's been previously dosed with a gene therapy, because the difference, and this is for some of the families, when we talk about, the neutralizing antibodies from kind of exposure to AAV you might have just in the wild or out just in your environment, the level of your antibodies that you're producing is going to be much different than if you've received a gene therapy, just because of the total amount of virus that you've received in that with that delivery. And this might be a good slide to stay on, because we had a number of questions kind of around some of the areas here. So I think maybe we can start with the mutation piece. So you have the exclusion of the deletions from 1 to 11 and then 42 to 45. Can we maybe break down the rationale for those two different groups of exclusions?
And Matt, maybe if you want to take these.
Sure. So when we look at the construct itself, there are areas of the construct that are going to be similar to these exons. And one of the concerns in the field is transgene immunity. And so we've excluded patients who might have deletions who in the areas that would be in the actual microdystrophin that we're talking about. So for people who have deletions including between exon 1 and 11, as well as those including or between 42 to 45, they would not be able to be part of the trial. Again, I know there's a lot of discussion in the field about this, and certainly as we move forward, that is something that we are also thinking about as well. But we do want to make sure that we're being first and foremost as a trial for safety, in terms of how we dose patients.
And then we had a question. So you said deletions within that range, or anyone who has a deletion of parts of that. Does that apply for things like duplications? If someone has a nonsense mutation, does the same rules apply here?
We have, for the trial purposes, we've only included deletions, not necessarily duplications or other variants that may be within that area. I think that would be also a discussion you want to have with the PI in a site as well, as each patient obviously want to make sure is safe.
Yeah.
And maybe to tag on to that too, I think that's been a really big topic for a while now, is understanding what are the right kind of conservative exclusion criteria as we learn more about these. The reason that we wanted to focus on deletion mutations specifically rather than any type of mutations are because if we think about kind of that natural immune response that the body has, it'll typically be against something that's foreign. And so when we have a patient that has a deletion in one of these regions and we are restoring that region of a protein, it's more likely than not going to be considered foreign to some extent. However, if someone has had a duplication or has had a different type of mutation, it may not be foreign. You may have actually had some level of exon skipping naturally that could have had a very small level of this being expressed, to which you may not have that immune response. So I think that's the important consideration and why we wanted to kind of move with that scientific basis for that specific type of mutation.
Great. And then again, for families who this might be newer to, or you're trying to maybe educate yourself a little bit more on this, the NIH did pull together a work group, and the companies who saw some issues with this transgene response in that earlier region of the gene, and there was some good data sharing. They did a webinar, I want to say maybe 2 years ago, but that is up on our Gene Therapy Hub on PPMD's website. So you can go back to that, and Karsten Bonneman and some other of the investigators kind of talk through what they were seeing, the SAEs that they were seeing, and kind of some of the rationale that we have. So that it's a good resource if you're trying to learn a little bit more about that. I did have a question relating to, well, actually, just to clarify. So I think maybe it was the previous slide, but you said this was an open label study. So as a reminder for anyone who's maybe unfamiliar with that term, this is not placebo controlled. Everyone who enrolls in the study is going to be treated with SGT-003. And I think there was a question around, are you going to be doing any functional assessments? And so I wanted to point out the time to rise you have, but can you maybe comment on the fact that that's day 540 as opposed to kind of maybe the 360 that we're used to seeing?
Yeah. So I'll start off with actually saying that the time to rise is one of the functional assessments we're doing. We are doing others. We also are doing others at different time points throughout. So that is not the only functional assessment that we'll be looking at. We've identified the day 540 as a key time point because that is 18 months. It is a little bit further out than some other studies have done. And that is really done to really try to maximize any potential to see change between what would be the natural history of the disease and potential changes that we might have after dosing. That's based off expert opinions, looking at natural history studies, as well as taking other data from trials that are both ongoing and in the past to try to make the best trial design going forward. So it is a little bit longer than I think you may see in other trials.
Yeah, and maybe just one thing to say is I think we need to learn from experience at this point. And I think what experience has shown us is that understanding just that heterogeneity that is going to be inherent to this period of time, being able to go out a bit longer could just give us that ability to detect a change a little bit better. So we want to make sure that we're evolving our study designs as we learn more as the field does as well.
Yeah, I think it's important to note that while we know the progression of the disease, the ability and time it takes for us to be able to maybe measure that difference at times may require changes to kind of our typical trial design in order to really show some of this. That's important. We had some questions around the kind of immune suppression strategy. So, I mean, maybe two things here. One, just can you describe what is being done with this increased prednisone dose? And then maybe just reflection on, we've seen across the landscape, and even in your 001, there were different kind of immune suppression strategies that were used, and how you kind of came back to using just prednisone at an increased dose for the immune modulation.
So the use of prednisone, I think since 001, we've also seen in the field as well. And so based off the preclinical data and the safety data that we've been able to see, as well as coming back to what it's been used otherwise, we felt that being on this increased dose of prednisone was going to be safe going into the trial as our marker. Patients do have to be on baseline steroids, as mentioned, for at least 12 weeks, and then coming in, they increase their prednisone. That's continued out for at least 30 days, at which point, per the protocol, it can be changed, weaned down at that point. That was done again because of the sort of close monitoring that we're going to also be doing during that time period, and being very careful and watching in for any signs or signals of concern. To date, as mentioned before, we have not had any serious adverse events in the trial in the first 3 participants. So that's sort of been the change that we've made going forward. We're also very cautious coming into with patients and making sure that they're in a good state when they start going to dosing.
And then just as a point of clarification, because we had a question come in around if someone was switching from Agamree to whether it's prednisone or Emflaza, the increased prednisone dose is on top of whatever stable steroid they're currently on.
Yes, they do have to go up to increase dose on top of the baseline that they have to come in on. I think that I saw a question someone answered early of they have to be on that for at least 12 weeks of the baseline of either prednisone or deflazacort before coming into the trial.
But they can be on either one of those and still.
Yep.
And then maybe we go to the next slide because there's some other questions still around some of these pieces. So one was just around age. So you have these 2 different age cohorts going from 4 to 12, and there was a question of if there's good safety data, kind of what would be your plan for potentially including older non-ambulatory patients or adults, or how Solid is thinking about that. Understanding, of course, this is your earlier phase 1/2 trial.
So certainly we want to be inclusive of the field as we think about things, but really the focus right now is making sure that the drug is safe and then that efficacious in the age groups we're looking at. The trial was, as Pat mentioned, it was very carefully designed using past experiences, both for outcomes, ages, things like that. So right now that is very much the focus, but as we move forward, certainly that is something that Solid's going to be keeping in mind.
Yeah, I think just understanding kind of where Solid came from, it's always been at our core to want to make sure that we're going to be getting to as many patients as possible. The reason that we had moved in this specific progression is that we do want to be conservative. We know safety is going to be the utmost importance as we can potentially expand. So our intention are to go to those different age groups as well. We're planning for it, but we want to make sure that we're doing that just in the right stepwise manner.
And then on the antibodies piece, so just, I guess, 2 things here. So we've seen across different programs, sometimes if you have any antibodies, that is exclusion, or maybe there's a threshold. So just, clarity on negative for AAV antibodies. And then maybe a little bit around kind of testing. And I think, families maybe are looking at private testing or maybe have been screened in other programs, but just to maybe for their sake, knowing that you have your own assay that would need to be tested for this, correct?
Yeah, exactly. I think the 2 pieces to it are when we're looking at a specific AAV, we do want to look at antibodies that would be most relevant to that AAV serotype. And then the second one is that antibody seroconversion, so changing from negative to positive, can happen within different periods of time. So for our study, we do require that an AAV antibody test happens in the screening period relative to dosing, because we want to make sure that that is an accurate assessment of whether or not there are antibodies to be able to then enable a patient to get dosed and to have that better confidence that they would be able to have that transduction and really the drug working. The saying that it's negative is really based on just our test that we're doing. So the specific test having its own cutoff for what is essentially a positive versus a negative. That's what's then used for this determination. So I think, other tests can have different ways to look at this from a certain titer may be selected because maybe you're allowing for a little bit more variability. I think as we also started here, what we wanted to just make sure of is that we're, again, using data to understand what is a positive result, what is a negative result that could have an impact on safety and efficacy. And that's where this test came in. So I think it is very important just to remember that, yes, it has to be within the context of the study for the specific test that we're using. Other antibody tests may be able to help guide, whether there may be positivity or not, but we can't tell until we use this test.
Thank you. And then just add another question on maybe just touching base again on kind of the exclusion of the other therapies and just the washout protocol for that first kind of screening for the study.
Yeah, one important thing that we wanted to do, and I'll let Matt also go into more detail, but was actually this is a change that happened from the first protocol version to this, is that acknowledging that there are changes in the field, we want to make sure that we're not blanket excluding patients who may be wanting to look at one of these different now newly approved therapies, especially that are available. However, because of the variability that can come from that, just in understanding what's the natural history of Duchenne progression over this time, we don't want that to be a potential confounder within the trial setting. So that's where we've allowed for this specific exclusion, but it would be a washout rather than a general exclusion. And Matt, sorry, any other details there?
Yeah, I think there's a lot that's been changing in the field, as Pat mentioned. And so we do want to make sure that we're also including patients that we can have that are safe, as we've heard a lot of time, more and more times, but also the efficacy data we're getting is good data and appropriate data. 12 weeks is thought to be enough that we can get a washout for these medications, and enough time that patients, we understand the logistics of screening, coming in and having to make these decisions. 12 weeks is still a lot of time to try to figure that out and go through that. So we felt this was a good balance, both from the scientific perspective, but also coming back to the community perspective of being able to allow patients to have that washout coming in and make that decision.
Thank you. And then to clarify, so there was a question around kind of the microdystrophin expression for how you're collecting that. So you're doing that through a muscle biopsy. Is that an open or a needle biopsy?
So it is through muscle biopsy. We are preferentially allowing a needle biopsy because it is less invasive. It is quicker. Someone who's done biopsies myself in my past life, open and needle. Sites are allowed to do open biopsies if that's what they prefer. So it's something to talk about as you go through the consenting process.
Okay. That's good for families to know. So have that discussion with the individual site that you're talking to about that. And then with some of the monitoring that you're doing, is there, I see, motor function, pulmonary function, any cardiac function being measured, whether serum or blood biomarkers or imaging?
There are. So we are doing both image echoes. We're also doing some serum testing throughout the study. At this point, the protocol is based on, again, on the safety piece. So we will be able to collect data for efficacy, obviously, with it, because it can be used for both.
And then there was a question, so this is a little ways back to, Pat, one of the earlier slides you had shown on kind of the microdystrophin construct that Solid is using and some of the work from Jeff Chamberlain and Dong Shen, but essentially asking around, with these microdystrophins having to make trade-offs because, as you said, the virus you can only package so much in there. So by selecting something like the nNOS, you're having to maybe make choices around other regions that you're not including. So just to maybe talk through again the rationale for that, that you think that this is the of what you looked at and we're using the best option and the potential benefits there.
Yeah, absolutely. And so I mentioned a bit about the structural components versus potentially the molecular components. And so if we look at the overall full-length dystrophin here, I think what's been really solidified is that the structural components have to be there to have a functional microdystrophin at the core of any sort of dystrophin variant. And really what those are going to be are the pieces that link the different membrane components. So your actual extracellular to the intracellular, and that's going to be the actin binding domain at one end, and then this dystroglycan binding domain at the other end, because that's the part that actually links up to the membrane. And so we know we have to have these core components. From there, in the internal piece of the dystrophin, what we have are some unique functions, but also a lot of redundancy. And so that's where you can see that each of these are different spectrum-like repeats, and many of them are still able to essentially perform similar functions as the others. So if you were to pull one out, another can compensate. And so what we've really seen, and this is a microdystrophin here, which is called microdystrophin 5 that came from Jeff Chamberlain, Dongsheng Duan, it's that we're packaging as much as we can into the AAV, but each of these components are able to result in that structural and molecular functional side. So if we were to say, add in another piece, another specific spectrum like repeat, we don't expect to see any additive benefit on top of that. And that's kind of similar to a couple of these others, but the unique part was this nNOS binding domain. So I think to your question, if we were to add in, say, a couple of different spectrum like repeats that may not otherwise serve a different function, we wouldn't maybe necessarily see additive benefit off of that. But the choice of using this specific nNOS binding domain is because it has that unique functionality that we would expect to potentially have that additional function.
And I think this is important too, just to highlight within kind of, for again, families maybe newer to the space and learning kind of around how we go through this process of testing products, that a lot of this, it's built on research that provides strong evidence that these are going to be hopefully, beneficial. And then you do the other preclinical work that you've done to kind of build that package to give us confidence. But ultimately, you do have to translate it and wait until we're in humans and running trials like you all are now, to really be able to tell if what we're seeing in these animal models is going to translate. And that is, one of the challenges that we always face.
Absolutely.
I don't see any other kind of questions coming in at the moment, but appreciate you kind of taking the time to go through and answer all of the ones that did come through. I do want to remind, families, whether you're watching now and, a question comes to you later, or if you're watching this on the recording, if you do have questions, please feel free to email us at PPMD and we'll try to get a response from the Solid team or make that connection. And again, information here for if you're looking for the sites to reach out to participate. There has been that expansion up to 43 participants for this study, so some opportunities there. But I think with that, we can wrap up, and I just want to thank the Solid team for joining us, going through that presentation and yeah, answering everyone's questions.
Thanks, Eric. Thanks, PPMD.
Thanks
Thank
so
you,
much.
everyone.